Have you ever wondered how a drug knows when to release its payload exactly where it's needed in the body? At ResolveMass Laboratories, we explore the science behind pH-responsive polymers, the smart materials that make targeted drug delivery possible. Today, let's dive into how these polymers work, the analytical science that validates their performance, and why they're reshaping the future of drug delivery systems. Let's start with the challenge. When we take an oral drug, it doesn't have an easy journey. It first encounters the acidic environment of the stomach, with pH levels as low as 1 or 2. For many molecules, this acidity can be destructive. Active pharmaceutical ingredients, or APIs, may degrade long before they even reach the intestines where absorption happens. The result? Poor bioavailability, reduced therapeutic effect and sometimes side effects from unintended exposure. To visualize this, imagine a delicate compound that needs to reach the bloodstream but gets trapped and destroyed in the stomach's acid storm. Traditional coatings offer some protection, but they're often not precise enough to ensure complete control over when and where the drug releases. That's where the idea of pH-sensitive materials was born. Materials that can sense their environment and respond intelligently. Researchers have already demonstrated how powerful this concept can be. In one study, famciclovir-loaded hydrogels released only about 25 to 35% of the drug in simulated gastric fluid at pH 1.2, but nearly 100% in intestinal fluid at pH 7.4. This dramatic difference in behavior proves that polymer chemistry can be designed to respond to local pH changes, acting like a molecular switch that activates at just the right moment. You can think of this like a relay race. The oral drug starts in the stomach, where the baton, the API, must be safely carried through acidic conditions without loss. But if the polymer carrier fails, if it opens too soon, the drug is degraded and never reaches its target. This is why understanding pH response and polymer chemistry is critical. The timing of release determines the success or failure of the therapy. So, how do these smart polymers actually sense and respond to pH? It all comes down to their functional groups, the chemical handles that interact with protons in the surrounding environment. Meanwhile, basic groups like amines, minus NH2, can also protonate, altering the polymer's solubility and shape. As the pH changes, for instance, when the drug moves from the stomach, pH 1 to 3, into the intestine, pH 6 to 7.5, the polymer undergoes ionization changes that make it swell, dissolve, or disintegrate, releasing the encapsulated drug. It's like a smart capsule that wakes up only in the right neighbourhood. A great real-world example is PMAA-PEG diblock copolymers. These polymers remain stable in acidic conditions, but swell dramatically at pH 7.4, releasing up to 90% of their insulin payload within 2 hours. For diabetic therapies, this kind of control is game-changing, protecting the insulin from gastric degradation and enabling absorption precisely where it's most effective. At ResolveMass Laboratories, we go far beyond just synthesizing these polymers. Our expertise lies in deep analytical characterization, ensuring that every polymer system performs exactly as intended. We use Fourier transform infrared spectroscopy to confirm functional groups and verify drug-polymer interactions. differential scanning calorimetry, DSC, to assess thermal behavior, detect drug crystallinity or amorphous state, and evaluate formulation stability. And dissolution studies that mimic the GI tract pH gradient to quantify controlled release behavior in real time. In addition, we leverage dynamic light scattering, DLS, and scanning electron microscopy, SEM, to understand particle size, morphology, and surface characteristics, key parameters that directly influence drug diffusion and bioavailability. Each of these tools provides a piece of the puzzle, ensuring that the polymer behaves predictably, safely, and effectively inside the body. When done right, pH-responsive polymers offer a remarkable set of advantages. They shield sensitive APIs from stomach acid. They enable site-specific drug release in the intestine. And they drastically reduce systemic side effects by preventing premature exposure. In controlled studies, such polymers have shown 90% release efficiency at pH 7.4, compared to only 10% at pH 1.2. A clear demonstration of their precision. But this is just the beginning. Future innovations are moving toward multi-stimuli responsive systems. Polymers that respond not just to pH, but also to temperature, redox potential, or even enzymatic triggers. With personalized medicine on the rise and advanced polymer manufacturing becoming more accessible, the horizon of drug delivery science is expanding faster than ever. In essence, pH-responsive polymers represent a paradigm shift, turning traditional drug carriers into intelligent therapeutic systems. At ResolveMass Laboratories, we're proud to be at the forefront of this transformation, combining material science, analytical chemistry, and pharmaceutical innovation to enable smarter, safer, and more effective therapeutics. Thank you for joining us on this journey through the world of smart drug delivery. If you're working on a novel formulation or polymer development project, ResolveMass Laboratories is your partner in custom synthesis, analytical validation, and advanced drug delivery design.